Um, in terms of the flora of the Bahamas, generally it's known that there exists a gradient from north to south in the Bahamas, a temperature gradient and a moisture gradient. And if you've had the chance to travel the islands of the Bahamas and pay attention to the bush, you can notice the shift in vegetation and in communities based on this gradient that exists. There have been persons currently studying this longitudinal gradient throughout the Bahamas and the Caribbean in terms of dry forest composition. My study more so wanted to look at the variation in species composition in dry forest communities in a very short, compact, longitudinal space, less than 1 degree of latitude. So, spending a lot of time in Eleuthera, and if any of you have visited Eleuthera, there's a very special place called Lighthouse Point. If you haven't been there, you need to go. Spending time on Lighthouse Point for conservation and recreational purposes gives you time to think. And I think I had read recently the ephemeral islands of the Bahamas by Dr. Campbell, and he wrote about the paleo-providence that existed back at lower sea levels during glacial maximas. And so one day I stood on the point of Lighthouse and looked over to Little San Salvador, which would be this little point here between the southern tip of Eleuthera and the northern tip of Cat Island, and had this vision of what would this strip of land look like back when it was exposed, when the Great Bahama Bank was exposed. What would that vegetation have looked like? And that just got me thinking and spurred my interest in studying the vegetation of these 3 points. And initially, this is an exploratory study to see what's there, in what amounts, in what assemblages, and see if there are any patterns we can pick up in this very small longitudinal gradient, if you want to call it a gradient. So my method is outlined as follows. I ran a number of plots, 75 in total, 31 in South Eleuthera and Lighthouse Point, 11 on Little San Salvador, and 33 on Cat Island. Using circular plots, 10-meter radius, surveyed for woody stems equal to and above 4 centimeters DBH, identifying them down to species. Also with 2 nested subplots of 3 meters and 1 meter respectively, 3 meters and 1 meter, surveying for shrubs, shrub vegetation that's less than 4 centimeters DBH, rooted in the quadrat, and the herbaceous vegetation not being woody and rooted in the quadrat as well. For plots, I recorded variables such as latitude, longitude, elevation, soil type, distance from coast, and distance from the road. There weren't very many roads in nearby some of my plots. So just to give an idea of the study sites, the pins indicate my plots that I completed thus far. For South Eleuthera, for Lighthouse Point, if you visited this point, you can see there's a very sharp contrast in the vegetation communities if you were to look at the leeward versus the windward side of this point. If you're on this part of the landscape, the substrate is mainly sandy. You have these rolling dunes that haven't solidified as yet, dominated by coastal scrub, dominated by Cocothrinax woodlands, and then transitioning into an a mixed coastal coppice, as I would call it, hardwood coastal coppice. Little San Salvador, a privately owned island by Holland Line America. Very little development. Usually cruise guests are confined to the southwestern shore. The rest of the island being dedicated as a wildlife sanctuary. This island being much smaller than the other 2 study sites, or the other 2 study islands. Vegetation communities different than on South Eleuthera and in northern Cat Island due to the island size and to the forces of the ocean coming off the Atlantic. This is a very sandy island, let's put it like that. Not very much elevation, not very much dark patches of soil, or substrate, or coppice forest, but it's very interesting being on Little San Salvador. In the northern tip of Cat Island, also a very large area. We have Orange Creek, which is here, This being mostly a coastal area, very flat. This point here, where the points are accumulated, known to traditionally be a farming area for locals in northern Cat Island. Land typically designated by locals as black land versus red land. So you would find that red lateritic soil. And cutting across the island, you pass through some sable wetlands, interior coppice, and out to the Atlantic shore again. So just some summary stats, and there's a lot of numbers from some of my plots, but just to point out, so the table on top represents a tree layer, shrub layer, and a herb layer. For Lighthouse Point, we had a total of 25 families, as well as for Orange Creek and Cat Island, 40 genera. For Lighthouse Point, 49 species. On average, about 50 trees per plot, and 10 species per plot, coming across 2 endemic tree species in the form of Thuinia discolor and, I'll have to check my notes. For Orange Creek, very similar numbers, 25 families, 39 genera, 45 species. The density was generally higher in terms of the number of trees per plot on average. Species per plot about the same, and 1 endemic tree species in my plot as Thuinia discolor. Little San Salvador, significantly less families, 24 genera, 24 species, 41 trees per plot, 9 species per plot, and no endemics encountered. For the shrub layer, you'd find that in Cat Island, it was really dense moving through some of those areas. And I don't know if any of you have moved. I'm sure a lot of us have spent time in the bush here in the Bahamas. And if you're not in the pine forest, it gets very thick, gets very dense, and in certain areas that are affected by whether it's the coastal forces or by human disturbance, most of the vegetation is about head height, very thick, very rough to go through. And so the shrub layer in Orange Creek on Northern Cat Island really reflected how intense, and also the tree layer. When we combine these numbers later on in my analysis, we'll see that the density of the plots in Orange Creek was definitely higher overall. Just to run some numbers, show some numbers again. So from my data collected from my plots, I calculated a relative basal area, frequency, and density, and using the average of that to get an importance value. And just to show preliminary results of what stood out in the tree layers. So in terms of basal area, which would be a measure of the width of the tree, as you measure the DBH, Bursera cimaruba and Coccoloba diversifolia being the most dominant in that category. In terms of frequency, that being in how many, which is the measure of how many plots did I find the species occurring. Acacia coreophylla, Metopium toxiferum, and Guapira discolor are at the top of that list. In terms of density, and that meaning how many individuals there are per plot, Coccoloba diversifolia, Metopium toxiferum, Bursera cimaruba pop out again. And in terms of overall species value, species importance value for Cat Island, Coccoloba diversifolia and Bursera cimaruba on top with Metopium toxiferum close behind. Just to show the other 2 study sites, in terms of basal area, Coccoloba diversifolia for Little San Salvador. Frequency, at the top of the list being Pseudophoenix sargentii. And in my other 2 study sites, being South Eleuthera and Northern Cat Island, I did not get any Pseudophoenix in any of my plots. Pseudophoenix covers this island. And in pretty much 10 out of my 11 plots, I got Pseudophoenix, which is pretty interesting, being that right next door on the 2 tips of Cat Island and Little San Salvador, I didn't find any. Now I know moving further up into Eleuthera, especially at the preserve, we find interior patches of Pseudophoenix sargentii. So I found that interesting. Density-wise, Coccoloba diversifolia and Reynosia pop up. And overall, in terms of importance values, Coccoloba diversifolia and Pseudophoenix sargentii. And back to South Eleuthera. Once again, basal area, we have Bursera cimaruba popping out with Reynosia in a close second. In terms of frequency, Guapira discolor pretty much showed up in in a majority of the plots. And if you walk the space at Lighthouse, you're going to hit a Guapira. If you close your eyes, you'll walk into one every few feet. Density, Reynosia Also very large numbers of darling plum in the plots in terms of number of individuals. But overall, in terms of importance value, Bursera cimaruba, Reynosia, and Guapira discolor pop up. Just in terms of moving through the space at Lighthouse Point, being very familiar with the area, I just did a very rudimentary map of the different vegetation types or community types found. And so the red indicates a Casuarina-dominated sandstrand formation or coastal scrub formation. If you walk that beach along Lighthouse Point, you notice the Casuarinas are very dominant there. Just behind that primary ridge, you have this series of rolling dunes, usually dominated by Cocothrinax argentata as a dominant tree species, but this is more of a coastal shrubland or scrubland, with the understory being dominated by Lantana involucrata, Cassia lineata, and other coastal shrubs. As you move to the opposite side of the big pond, this blue area indicates what I would call a mixed interior hardwood coppice. And although this is all one color blue, this is hardly a homogeneous community. You can move, for example, if you were in this area, you would find large stands of Guaiacum sanctum, our national tree, Lignum vitae. As you move from this tip into this blue region, there's a transition zone from an exterior coppice or coastal coppice to more of that interior coppice. And basically, as I've been going through and doing the plots, Bursera cimaruba has been dominant. Reynosia septentrionalis has been common, but it's it's a hodgepodge. It's a mix. It's a shifting mosaic of communities. And so, just to show just a few images, this shows what the typical coastal shrubland dominated by Cocothrinax argentata looks like on that rolling dune system. As you move closer to the pond, to higher elevations, it turns into more of a coastal coppice with Xanthoxylum flavum dominating along with Bursera cimaruba. And as we move to the opposite sides of the pond, it gets to more of a mixed coppice where you find epiphytes, Acacia coreophylla. The substrate is darker and more solidified into rock. Just because I like to have pretty pictures. Is some flowers that images I picked up along the way. And just to close, in terms of the future analysis, so this has just been preliminary data. I plan to use ordination and clustering to recognize distinct assemblages of species and see how they relate to one another. I've been doing some preliminary running of R. I think R is running right as I speak. It seems as though elevation and distance from the coast are two strong indicators of species assemblages, and so we're looking forward to completing this part of the analysis, not only to understand the variation in this relatively short gradient space, but also to compare to larger studies that look at the gradient across the Bahamas and also across the larger Caribbean. And so on that note, I'd like to thank the Bahamas National Trust for inspiring me, for giving me a job, and hopefully give me a job when I come back. Miami University, who's funded for me to be here and funded my research, the management and staff of the Leon Levy Native Plant Preserve, the summer interns, folks at the Best Commission, Ministry of Agriculture. Who am I missing? I would say Custom and Border Protection, but they took my specimens, so we're not going to thank them. Other than that, thank you very much.